Hi everyone. This tutorial is going to move us into our study of the biological macromolecules, and this will be the focus of the rest of this first part of Unit 1. So if you want to follow along with your notes, you'll want to be looking at your Biochem Note 3. So in this look at the biological macromolecules, we need to begin by thinking about the atom carbon. Carbon forms the backbone of all the major biochemical molecules. And the reason why it's a great element to form the skeleton of these large molecules is because of the fact that it has 4 valence electrons. Having 4 valence electrons allows the carbon atom to form 4 bonds. And these bonds can be separated around the outside of the atom to create the tetrahedral shape. By having a tetrahedral shape, you can expand upon this atom and form a huge diversity of architectures of molecules. Carbon can form single bonds, double bonds, and even triple bonds with other atoms, particularly with other carbon atoms. It likes to form bonds with hydrogen and oxygen, phosphorus, nitrogens, sulfurs. These are the main elements that make up our biological molecules. These carbon molecules can also form long linear chains. These long linear chains can branch and form even longer chains. And the chains can also fold back on themselves and form a variety of ring structures. So as we work through these biological molecules, you're going to see all of these different architectures of molecules. In order to understand how carbon-based molecules can form these large, diverse groups of molecules, you need to have an understanding of something called functional groups. Functional groups are groups of atoms on molecules that are highly reactive. They're found on the sides of a molecule, the ends of a molecule. They may only be made up of 2 atoms, or they may be made up of dozens of atoms, but these small groups are very reactive. So this allows smaller molecules to link together through chemical reactions in order to form these larger, more complex structures that we see in living cells. Here's a graphic that shows you some of the common functional groups. I don't expect you to have to memorize all of these different functional groups and their structures, but as we work through the macromolecules, you're going to see these groups come up over and over and over. And after a while, the common 4 or 5 that we'll talk about a lot will become just secondhand knowledge to you. So don't get stressed out about all of the structures on this particular slide. But I just want to introduce you to some of the really common ones. This is also in your textbook, so if you're having trouble seeing this slide, just take a look in your book at the list of functional groups. The first one that you see at the top is the hydroxyl group. So a hydroxyl group has an OH, an oxygen and a hydrogen. And the compounds that have hydroxyl groups are referred to as alcohols. Hydroxyls will be very important in our study of carbohydrates and lipids in particular. The next group is called the carbonyl groups. Carbonyl groups have a carbon that is double bonded to an oxygen. So you can see that over here. And this creates 2 groups of organic compounds called aldehydes and ketones. The third group, right here, is the carboxyls, and you'll see this group a lot when we go through the next week or 2 of classes. The carboxyl groups have a carbon double bonded to an oxygen, and then also attached to the carbon will be a second oxygen. In the non-ionized form, that'll be an OH, and in the ionized form, it'll be just a charged oxygen group, like you see here. I like to refer to this as the COOH group. That's how it looks most of the time, and it will be attached to a carbon on another molecule. The next group are called the amino groups. The amino groups have a nitrogen in them, so that's how you can always spot them. They're the only ones with nitrogen. They'll either have 2 hydrogens with them, or they'll have 3 hydrogens when they're in the ionized form. So it'll either be an NH2 or an NH3 with a charge on the end of the molecule. The next group is the sulfhydryl group, also referred to as thiols, and we're really only going to be looking at the sulfhydryl groups when we study amino acids with proteins. They play a really important role in protein structure, and so that's where you'll see them in a couple of days. Phosphates are a phosphorus with 3 oxygens around the phosphate. Sorry, I'm going to erase that because I realize I'm spelling the word poo. More accurately, we would have PO4, or in some forms it's a PO3 group. So these are the major functional groups: the hydroxyls or alcohols, the carbonyls. Some people call this a carbonyl, and it's just a carbon with a double bonded oxygen. The carboxyls, which is a COOH group, the amino groups that have the nitrogen, the sulfhydryl that have the sulfur, and the phosphates that have the phosphorus in them. Just try to familiarize yourself with these, and you'll see these a lot in the future. Okay, so moving on, we'll now start to look at the major 4 groups of biological macromolecules. These are the big molecules that make up all living cells. And you've heard of them all before, except maybe the 4th one here. But we have carbohydrates, lipids, proteins, nucleic acids, and nucleic acids include DNA, which you've all heard of before, RNA, this will be something new, ATP, I think you may have heard about last year, and NADH, a molecule you'll learn about in the next unit. So the major macromolecule groups, and we will begin by looking at the carbohydrates. Now, some key terms that you're going to want to be very familiar with as we work through the macromolecules include the functional groups, which we've already talked about, monomers, and the word mono, or the prefix mono, means 1. So monomers are molecules that are made up of only one molecule, one main base group to the molecule. Polymers, poly is a prefix that means many. And so these are large molecules made up of many, many, many smaller repeating molecules. So polymers are made up of monomers. And the last 2 key terms are condensation reactions and hydrolysis reactions. And I'll be covering these towards the end of the video. Actually, my mistake, we're covering the condensation reactions now. I'm trying to follow the order of what's in your note that you're currently looking at. Okay, so condensation reactions. If you want to put 2 monomers together, 2 smaller molecules together, your cells conduct a condensation reaction. And just so you know, condensation reactions are called by some people as dehydration synthesis reactions. And I actually like this name more because it makes more sense to me. Dehydration. When you're dehydrated, you're missing what? You're missing water. And synthesis means to create something, to make something. So in a dehydration synthesis reaction, you're building something, you're making something by losing water. And so you can see that right here. So here's a reaction between 2 amino acids. In amino acid number 1, over on the side, we have this carboxyl group. And in the carboxyl group, there's this OH. In amino acid number 2, we have the amino group on the end of the amino acid, and it has a hydrogen. Well, what do you get when you put a hydroxyl with a hydrogen? What will that make? It makes water, H2O. Okay, it's actually, what it is it's an O with its H, and then it attaches another H to it, and that's why we see water here. So by the removal of one water molecule, it creates a bond. The bond happens between the carbon right here and the nitrogen right there, and we get this bond. And that's what links these 2 amino acids together in protein structure. So that's called a condensation reaction or a dehydration synthesis reaction, and we're going to see these in the carbohydrates, the lipids, the proteins, and in the nucleic acids. This accounts for the building of larger, more complex molecules. Now, oppositely, your cells will do hydrolysis reactions. In a hydrolysis reaction, water is added in to the reaction in order to break the bond between two smaller molecules, between two monomers. So here's the example of taking sucrose. So you've just eaten something that has sugar in it. Your digestive enzymes will use a hydrolysis reaction to break the bond. Oops, sorry. Oh, I don't want to move this picture around. I just want to draw on it. Here we go. To break the bond between the two monomers. By doing that, it creates a hydroxyl on one end of the one monomer, and it adds a hydrogen to the oxygen of the second monomer, and so we have these two hydroxyl groups on our two monomers. They're now separate. Sucrose has become a glucose molecule and a fructose molecule. So we do this when we are digesting larger molecules into smaller molecules. So this is called a hydrolysis reaction. Hydro, no, I don't want to do that. Just want to write here. Hydro means water, and having some trouble here. Lysis means to break. So hydro means water, lysis means to break in Latin. So hydrolysis reaction uses water to break molecules. Get it? Okay, so now I'm into the carbohydrates. I've pasted a collection of photos here of various things that have carbohydrates related to them. So we have a crab, some glucose powder, a picture that has grains and potatoes and pasta and wheat, rice. Over here we have, this is a picture of plant cells, and particularly I'm looking at the plant cell walls. Here we have my favorite athlete, Usain Bolt, great Jamaican. He's pretty muscular and he needs lots of energy for running. We have milk, and we have good old table sugar over here, white table sugar. So we're going to talk about these different kinds of carbohydrates that are related to all these substances that you see here.